Yeah, I'm not used to doing in-person things anymore. I don't know if any of us are, but it's really great to be back, and I'll talk to you a little bit through some of this. I think 3 years ago when I was here, probably no one had heard, or very few people had heard the term kratom or kratom. Now I think it's hard to not drive past the vape shop and see a neon sign for it, or at least flags flying outside advertising the name of it. So it's grown in popularity just in the last few years tremendously. I think some of that is the funding effect, the effect of the government taking interest in this. It must be good if the government's interested in it, so people are interested in it even more, right? But I show you a picture here of trees growing in Malaysia. This is a pretty typical look at what a farm is there. I didn't show anywhere location-wise because it is illegal in Malaysia to grow it for sale. It is legal for people to have their own and consume it, but it's not legal to sell in the streets. But I'll show you that they do that too very well. So first, okay. First things first. I know this is small and not many people can see, but we will first tell you that this is not work done by me. This is work that I've had the real honor of chairing and sort of directing a bunch of people to work on. We've had over 30 people involved in this project in the last few years. Many of them are listed here, so there's been a whole group dedicated to chemistry, isolation of natural products from the plant, structural characterization and elucidation of that. I've had some very talented PhD scientists working in that area, along with some students from the pharmacy program at the University of Florida and visiting students from the University of South Malaysia that actually were bringing their own products over from Malaysia to work with us on as well. We've done a lot of receptor binding studies, so some of the in vitro pharmacology, lots of in vitro and in vivo pharmacokinetics work that's really been monumental in helping move this whole project and understanding forward. And under that line, at the very bottom of that list, is Ali Sinetra, who's a Rowan grad alumnus, and she started in our graduate program a couple of years ago and has been doing very well. And so if anybody's interested in coming to Florida for graduate school, talk to me. We have a good track record so far, so we'll keep that rolling if we can. Behavioral pharmacology, I get my feet dirty in this area, but I trust those that have been trained very well in this area to work with and take the helm of those projects. Our main collaborator, Lance McMahon, left the University of Florida a couple of years ago to take up the vice president for research at Texas Tech Health Sciences. And so he is now there. We're still collaborating and working together. And then we've worked with some folks like Scott Hemby, who's at High on drug abuse, but we've also had a tremendous amount of kratom vendors, so people that are actually in the industry that have come to us and work with us to provide product, to give us really what's out there being sold and look at and interrogate those products to understand them fully. Tons and tons of student workers. I don't really have the opportunity to tell you how I got my start, but it was because a faculty member asked me to come work in a lab one day, and I started working in a lab, and now look where I am. So I was going to be a pharmacist. That didn't happen to the full extent. I did practice for a number of years, but I kept realizing that my interest really lay in the research realm and really lay in the educational realm and trying to get information out to as many people as possible. And so I feel this plant has sort of given me that opportunity to come out and speak about it as well. So we started working on this plant in 2005, before it was even on the radar of the Drug Enforcement Agency in the United States, or the FDA. And so in some ways I feel guilty, in some ways I feel really glad. Because the guilty aspect is that we kind of helped promote and introduce this this material. The happy aspect of it is that we're able to study it. We're able to add some science behind a lot of the anecdotal evidence that's been presented or given out, some of the misinformation that has been given out, particularly to regulatory agencies like the DEA and the FDA, and help really construct policy around this. And that was underscored a little bit more this past year when I was able to speak to the World Health Organization, and they were considering an international ban on this, which ultimately they decided not to do a deep investigation into this because of two reasons. One, the traditional cultural use of this plant, which I'll talk about here in just a moment. And then also the fact that the emerging evidence based on the science that's coming shows that there's potential medical and therapeutic benefit coming from the work that we're doing, along with many others around the globe. We're not the only ones by any means working in this, and we weren't the first either. But I'll just take you on the journey, and I just realized that the biggest typo that somebody can make when talking about a plant is putting the genus capitalized, which it's supposed to be, and the species capitalized, which it's not supposed to be. So just to tell you that full professors always still can make mistakes. So Mitragyna speciosa is the botanical name for this tree. It was given the name Mitragyna in this genus because of the shape of the leaves. The leaves are shaped like a bishop's hat, or what's known as a mitre. And so if you actually take that leaf and turn it backwards clockwise 90 degrees and imagine a face underneath where the stem is, that's your bishop's hat, if you will. It is a flowering evergreen tree in Southeast Asia, and it is found primarily at the border of Malaysia and Thailand. We believe that's where the species sort of originated from. It now spreads all throughout Southeast Asia. Most of the material that's coming into the United States is being grown in Indonesia, but it has been accounted for all throughout the Southeast, even over into Eastern India. And so we know that there's a lot more being grown these days than where it originally started. We are fortunate to be working with universities in Indonesia, Thailand, and Malaysia. So we have sort of the whole main area of where it's being grown, where we're collaborating with groups that have access to fresh material there. And it has various names. We hear it called kratom most of the time in the United States. Kratom is a totally United States made up name. You say kratom in Southeast Asia and they'll look at you like they have no idea what you're talking about. They call it kratom or kitom or yak yak. So kratom is like totally foreign word to them. But those are the actual terms. You'll hear me call it everything because I'm sort of in the philosophy that tomatoes, as long as we know we're talking about this product, I think we're okay. There are about 40 different alkaloids that have been isolated from this plant. Many of those are in minor quantities, and we've been fortunate enough to have large volumes of material to isolate from. And so we have some studies that have been done on alkaloids, and I'll talk about some of the pharmacology, where we're the only ones that have done it because there's just not enough of it to isolate in large amounts. Kratom use in Southeast Asia looks a lot like this picture. Kratom tea in Malaysia particularly is brewed with fresh leaves. So in fact, almost every house in the rural area of Malaysia, northern peninsular Malaysia, right on the border of Thailand, almost every house in that area has a tree growing in their yard. And this is a traditional consumed product by almost everyone every day in that region of the world. So they'll pick fresh leaves off the tree in the morning, they'll cut those leaves or rip those leaves, put it into a pot of boiling water, they'll steep that for 2 to 3 hours. And then they will ladle out the beverage, allow it to cool, or they'll drink it warm. But they generally package them up in either a water bottle like that or in a plastic baggie like you see here. And when I first saw that plastic baggie, I thought this is a really weird way to package something up for the whole day's use. But then as I walked around Kuala Lumpur in Malaysia, the main city, which is incredibly modern and beautiful, I saw a lot of people walking around drinking bubble tea and other drinks out of plastic baggies like this. And I was like, oh, that's like more environmentally conscious than something like this. But anyway, that was just sort of the sort of normal one-day use packaging is either a bottle or a baggie of the tea. And so they would use that tea 3 times a day. Most individuals, they'll take a little bit in the morning, a little bit in the afternoon, and the rest in the evening. On the weekends, they may increase the evening dose on Friday or Saturday night when they're hanging out with their friends or their neighbors, more as a socially relaxant drink. And so it does have this stimulating property, this ability to really improve work efficacy in that hot, humid, tropical climate, which, by the way, happens to be very similar to what Florida's climate is. And we'll talk about that in relation to growing the trees as well. And so a lot of this can be looked at as stimulant at lower doses, essentially like what we drink coffee for, right? Get us up, get us moving. I think this is sort of their coffee in that area of the world, where they'll utilize this throughout the day and keep going. But you can, if you increase the dose, get into euphoric or sedative type effects, which are probably related to some of the opioid activity that's within the plant. And that's been the big concern around the United States, at least. The other picture that's there in the bottom sort of center is a roadside stand that sells Kratom Tea. And so you can't really see any of the people, that's purposeful. But if you can work your way inside that, it's right on the roadside. You can drive your car right up to it, jump out, buy you a bag of this stuff, and be on your way. And remember, I said it's illegal to do that. And so the police usually just keep driving by it. They're not that concerned about it. From time to time, they'll make an example and give somebody a fine, but it's really not too bad. Interestingly enough, Thailand made Kratom illegal in the 1940s because it was cutting into their opium tax. And so there was an interesting legal political play that was made, and they ordered destruction of all the trees in Thailand. And I don't know how many of you are familiar with kudzu in the southeastern United States, but kudzu is now being eradicated. But 10, 15 years ago, if you would have said to somebody, destroy all the kudzu in the United States, it is like an impossible thing to do. It grows 3 meters a day, 3 feet, sorry, 1 meter a day. These trees grow very rapidly and very quickly from seed. And so it's almost impossible to get them down. The big thing that attracted our attention and most of the attention is that the tea is used to reduce opioid withdrawal. So a lot of these laborers would drink the tea throughout the day normally, but many of them were also using opium. So not a prescription opioid, but the real natural opium product. And when they would run out of or not have access to opium, they simply would increase their consumption of these teas and not go through withdrawal. And so this was attractive from our standpoint at looking at something that might be able to utilize in the arsenal of things to treat the opioid crisis. So that's really where our entrance comes into this. More recently, just a couple of years ago now, we published a paper that showed poly drug users that are using methamphetamine as well as opiates in Malaysia have been able to reduce their intake of methamphetamine by using this product as well. So then the question always comes up, is it the chicken or the egg, right? Like, is this replacing opium because it's just as good as opium? or is it really something that's beneficial and helpful? And I think that's where a lot of the question in the public is to this day. So the predominant active ingredient is mitragyna, and this is a coinanthine-based, indole-based alkaloid that we'll find. It actually exists as two sets of diastereomers in the plant. So there's 4 molecules that are identical in their makeup in the plant. One of those is named mitragyna. It has been reported to be the most abundant of the alkaloids in the plant. That was based on a paper that was published many, many years ago. And it was based on a sampling of 3 trees. Everyone since that paper has come out has basically put their weight on the fact that this is the active and predominant alkaloid. So I'm going to show you today that that may or may not be the case, but there's a lot of other bioactivity in this plant. There's a lot of other interesting pharmacology in this plant other than what this compound is giving us. So if you think that this is the main player, which many people still believe, and I'm not going to argue against that, there are 3 other chemicals that just have differences in stereochemistry in the plant. Some of those are almost as abundant, depending on the chemotype of the tree, and I'll talk about that in a moment. You may actually have higher amounts of certain molecules than you do of mitragyna. In some cases, we've seen some trees that are producing almost no mitragyna at all. So it all depends on the tree and what the market interest is. The market interest in the US right now is for a concentration of mitragyna to be at a certain level between certain percentages of weight in that leaf material. And that's what vendors in the marketplace are looking for. And so that's what we see in the marketplace in the United States right now. So bringing us a little bit closer to what is kratom use in the US look like, it's a very different picture than what you just saw on the previous slide, right? There's no more tree, there's no pot of water, but there's a lot of products. And so individuals are buying leaf material that's either crushed and dried or very finely ground. They can utilize those to make teas. They can chew those powders. They ingest it various ways. I've even heard of people skin popping kratom powders, which I would not ever tell anybody to even think about doing. This is just a bad idea. But you also see that there's capsule forms, there's tablet forms out, there's energy shot forms, concentrated extract forms out, and what else? Oh, and then there's just various packaging. Some that look very, very pharmaceutical or very nutraceutical based that will say, you know, Mitragyna speciosa, or will say Megong, whatever type of kratom material it is, or Indonesian white, or something that sounds much more like a botanical. And then you have products out there that are marketed as things like Foria or Vivazen that sound much more like, hey, this is going to feel good if I take this, and it enhance my mood. Of course, nobody can make any claims therapeutically about these. Nothing has been evaluated or approved by the FDA. So all these are being sold in the marketplace as either not for human consumption or for just a dietary supplement. It is sort of regulated in the food space or the nutraceutical space right now, if it's regulated at all. The FDA's official stance is that it's a non-regulated opioid, and the FDA wants to see it sort of, you know, controlled and regulated in some way. So we're working closely with a lot of the regulatory agencies to talk through the science to understand where we should be, because we also realize the blunt fact that you can see on this slide. There's about 2,000 metric tons coming in every month to this country, which, by the way, the FDA has an import ban on this material into the United States. So all this 2,000 metric tons that's coming in, which is probably at least double this now, because the use of kratom and the popularity has soared during COVID, as have overdose deaths from opioids. But what we realize is that just by doing the typical dose calculations, there's more than 15 million users of kratom in the United States. That number is probably even larger. It may not be consistent users. It may just be that many people have been exposed. We're starting to even get granular into learning which states have higher use rates than other states. Florida just happens to be one of the places that is very, very popular. But we will learn more and more as we go. So how is something that's banned from coming into the United States in this much quantity every month? It is brought in under the auspice of being sodium alginate. Anybody know what sodium alginate is?
For molecular gastronomy.
For molecular gastronomy, this is true. So it's actually seaweed byproduct that's used as a thickening agent in the culinary industry. And many seaweed products are brought in all the time, and nobody's going to open up a box and see a bunch of green powder in there that's labeled sodium alginate and guess that it's not sodium alginate. So a lot of it's coming in that way. From time to time, you'll hear about a seizure that's taken place at the ports of entry, but it's coming in at a good rate. And once it's in the country, there's no restrictions in most of the country in terms of sales. It is now illegal in 6 states. Those are listed at the bottom of the slide if you can read it. But that's Alabama, Arkansas, Indiana, Rhode Island, Vermont, and Wisconsin. There's several cities and several counties throughout the rest of the United States where it has been banned or outlawed. And interestingly enough, in many of those bills, it's listed as a synthetic. So it's hard to put my head around how it ended up lumped in with the synthetic cannabinoids and synthetic fentanyl analogs, but it's this natural product. So I'm trying to understand and educate and get information out there that's proper is really key. I'm not here to condone sales of this product in an unregulated marketplace at all. We're here to understand the fact that it is out for sale in an unregulated marketplace, and what is it doing to people that are ingesting it? What are potentially safe levels or unsafe levels of this product, and how can we help to influence safety in the public and the policy, and really do what the FDA wants to do? So the question comes down to it, is kratom a threat or is it a therapeutic? And depending on who you talk to and who you talk to within certain agencies, they may tell you it's a therapeutic or they may tell you it's a threat. I will say that a lot of this depends on the dose. Just like any drug or any substance, it can be toxic if you get to a certain level. Water can kill you if you drink too much too fast. Not by choking, water intoxication. So there's a lot of things that we live around all day, every day, that can be just as deadly. Anecdotally, again, this is really used for chronic pain, mood elevation, opioid use disorder. I receive emails from users constantly, always sharing their story about how this has changed their life, how it has gotten them off of opioids, how it has gotten them off of the couch, how it has made them re-engage with their families, getting their jobs back, getting their energy levels back, and eliminating their pain. And so it's interesting when you hear about it used in the sort of context of that versus other individuals that email me and say, this is the greatest stuff if you want to get high. You just take all this much, you just keep taking it until you feel it. And I'm like, how can people do this, right? But there are products in the marketplace that are heavily concentrated extracts with 45 times more alkaloid content than you would normally experience from just leaf material. And then these individuals are downing 4 or 5 extract shots at one time and ending up in situations where they're contacting poison control centers or in the emergency room. And if you talk to most physicians, they think Kratom is a bad thing because all that they've seen in the medical literature and in their emergency departments are people that have done what I just described. Overindulged in a product and ended up in that emergency room to be seen for some issue. Many of these are in combination with other drugs. Many of these have led to severe problems with that individual ending up in those situations as well. So there's always the question of adulteration of product. And we've published papers now on what we thought was adulteration by increasing levels of certain alkaloids within the plant. That was before we learned how difficult it was to actually get some of those certain alkaloids to high concentrations. So I'll talk a little bit about that in terms of the chemistry of those and pharmacology. But we have seen products and analyzed products where they were laced with hydrocodone or morphine. And so there is still some concern and some scary aspect of this wild, wild west market that's not regulated, which you really don't know what you're buying when you buy some product like this. The other thing is that kratom has been claimed to be severely addictive, and so there have been several reports in the literature, and the CDC as well as the FDA came out and talked about this being linked to several deaths, kratom products, several deaths. And that USA Today story that I highlight on this slide is an interesting one because the headline reads, herbal drug kratom linked to almost 100 overdose deaths, CDC says. If you read that article at the very end, it says, in almost all cases, fentanyl was found in the person's system. So it's been an interesting media story about what really is the problem or what isn't the problem. And we're trying to understand how do those things interact with each other? Is there drug-drug interaction problems? Most people that are consuming kratom to get a recreational use out of it, more than likely are using other things at the same time. And so how do those things all play together? And do they play nicely? As far as the addiction goes, I work with several addiction physicians, and they utilize kratom off-label because it doesn't have any label or claims, but they do utilize it in patients to treat them for opioid dependency. However, they also started coming to me and saying, hey, we're starting to see people showing up with kratom addiction. And what do we do? How do we treat it? I said, I don't know. We work with mice and rats. I can't tell you what to do with people. But it's led to some really interesting questions and really finding out how people are using it, what patterns are using it, how they're getting into the situation, and what can we do? Because I'll show you that the pharmacology is not just opioid related. And what our concern is can we treat these people the right way? If it's not all opioid and we then put them on something like buprenorphine that is a pure opioid, are we just taking them off of one substance and not putting them onto the pathway to opioid addiction? So it's a very fine balance of trying to understand what's going on. And as I mentioned, the United Nations WHO already rejected the international ban on this. So what's the story and what's going on? If we look at the case reports, I would say that very few, if any, deaths are attributable to kratom alone. The reason I say if any is because we may not know what we were looking for. So if Mitragyna is the biomarker that they're using in forensic analysis is the only substance that shows up, it's because it's in the database and they're looking for it. If it's a synthetic fentanyl analog or some other medication, some other illicit substance that was in that mixture that they ingested that isn't in the database for forensic analysis, it's going to be missed. And that could easily contribute it. And of course, we want to be a society that has an answer to everything and say, okay, this is it. This is all that's there. We're done. We don't know that that's really the case. However, there are 2 cases of kratom-related death. Both had other substances in their systems, but they had over 100 to 300 times the normal blood level of what a typical user in Southeast Asia would have in their blood. So it's hard to say what's going on. We do know that that major alkaloid is a partial mu agonist. So this works on the same receptor as morphine. It gives about a 40% maximal activation. So it's a weak, really partial agonist. And that's just demonstrated there with those agonism curves, where in black, you see Damgo, which is a typical mu opioid reference standard full agonist. And in red is Mitragyna that cannot produce a maximal effect in that assay. We also know that Mitragyna was really of high interest because it's a biased signaling agonist. So it has no recruitment of beta-arrestin. And if you don't know what beta-arrestin is, that's okay. We don't really need to go into detail. But the excitement around that compound is that, or not recruiting beta-arrestin, is that opioids that don't recruit beta-arrestin in that signaling pathway seem to have a better side effect profile. So less respiratory depression, less constipation associated with it. So it's been believed that if we can have a selective ligand that only works through the G protein and doesn't recruit this other protein called beta-arrestin, that we should have a safer profile. Now, two molecules, one that has been approved by the FDA, TRV-130 or Oclaridine, is now on the market. And it was originally rejected by the FDA, but they are this biased signaling pathway. They don't recruit beta-arrestin. And they came out with this whole idea that it was going to be so much safer than all the other marketed opioids. But at the end of the day, the FDA and all the evidence showed that this had no different safety profile than any opioid on the market. So the whole theory of this beta-arrestin exclusive pathway kind of got brought into question. There's still compounds that they claim are even more selective that are in development. And so we'll see where this story takes us. But for right now, we're not entirely sure that this is giving us the safety profile that we see. So what about therapeutic potential? I've already mentioned that we're looking at kratom for opioid detoxification or treating opioid withdrawal. Also for medication-assisted therapy, so something like a market competitor for methadone or buprenorphine. And what we've seen, at least in self-reported data in the literature, and now there's actually clinical trial data that was shared at the College of Problems of Drug Dependence this year, is that the subjective opioid withdrawal scale, in other words, the self-reported withdrawal feelings, is very mild. So very similar to what's coffee. And we hear that a lot. We hear that a lot. If you talk to somebody who's battling kratom addiction right now, they're going to tell you it's worse than coffee, for sure. So there's probably some spectra in all of this. We do know that kratom as a plant has multiple mechanisms of action, and I'll get into the chemistry, I promise. And we really are trying to understand all those different mechanisms. But we know for sure it has adrenergic activity, serotonergic activity, and opioidergic activity. And it may be the combination of those things that really makes it interesting. So lacking a standardized product has really helped us do any rigorous clinical trials to evaluate that. Whoops. Come in my good partner now and collaborator, Brian Pearson, at the University of Florida's Institute for Food and Agricultural Sciences. We now have our own forest of Mitragyna speciosa growing in Orlando, in greenhouses in the fields. And what we've learned from this is that whole tree sampling issue I mentioned before. So we're seeing trees that produce around 2 to 3% Mitragyna in total alkaloid content, not 66% total Mitragyna in alkaloid content, up to about 45% Mitragyna in alkaloid content. And so there's definitely a difference. And I also like to look at this as almost medical marijuana versus hemp. And so what you have is a high producing Mitragyna chemotype and a very low producing Mitragyna chemotype. The very interesting thing, and I can talk about this for hours, I promise I won't. The very interesting thing is that low producing chemotype, all of those plants have been donated to us from people who have been using their own homegrown plants and say it works just as well, if not better, than the products they were buying in the shops. Now, we don't know if that's a placebo, we don't know what it is, but it's enough noise from enough different providers that there may be something there. And so I go back to what I mentioned earlier about Mitragyna maybe being the thing, maybe not being the thing. This is a variety of the alkaloids that we have isolated, not a complete list, but the ones that we've isolated enough of to get full binding profiles on and understand pharmacologies with, also perform full pharmacokinetic analysis and behavioral pharmacological analysis, including addiction potential for each of these compounds. And what you can see essentially across the top row are 4 molecules. The 2 in the middle are actually just a reduced hexacyclic double bond from each other difference. But other than that, they are those isomers or enantiomers I was talking about, diastereomers I was talking about. There is another one that's not listed up there because we've just now isolated enough of it to characterize. It's in such low amounts of the plant material. But all of those molecules, if you were to use an insensitive method, like just UV UV detection on a LC system, these will all come out with the same exact wavelength chromophore. Unless you really knew you have 4 compounds in there, you would just say this is 1. So it's very possible that those original reports of Mitragyna being 66% of the alkaloid content were because they were looking at all of those compounds together. Then, if you think about that from another standpoint, once these compounds start to get metabolized, you're generating the exact metabolites chemically 4 times over. So we're getting demethylation of Mitragyna's indole ether there, and that happens to be also a very active natural product as a metabolite, but we also get that on each of the different isomers. And so if we're looking at metabolite quantification, now we have to make sure we have baseline separation of those 4 metabolites in addition to all the other oxidative metabolites that are going to be generated. So it turns into a real mess. Anyway, I put the historical numbers up here for what the ratio should be. And we realize that these ratios are not really there. I'm not going to go into a lot more detail just in the essence of time around too much of the structure activity relationship, but I would point out one last thing, in that all of these alkaloids contain what we call beta-methoxyacrylate moiety at the bottom of the molecule, and this is conserved through every single alkaloid that we've isolated from the plant. The question was, what is that doing? Why would something that looks like a Michael acceptor or a reactive moiety on a molecule be present in so many different alkaloids from the plant? And also something that's an ester, a methyl ester, is pretty susceptible to cleavage very quickly in the blood. This beta-methoxyacrylate is rock solid stable. It's not very reactive. We have not detected the addicts with it. And we see very little of it, although it is one of the major metabolites, the acid, it's still pretty stable for a long time in the body as the parent compound. So we don't know what else is going on there. It is related to another functional group that we see in some natural products that are related to antifungal activity. And we think that the trees are growing in swampy, wet environments, and they may be just exuding these different chemicals as antifungals so they don't overcome my fungus. I don't have any evidence to say that that's the case, but that's kind of our current hypothesis. We did run these compounds through all these targets, and I'll just quickly orient you to what's on this slide. This is a screen against 82 different drug targets. What comes back is when we displace 50% of the radioligand at 10 micromolar concentration, we get an X on the graph, so to speak. And that just tells us that it's interacting with
every year.
Then we do a secondary screen at 100 nanomolar, and we look to see if we
I'll do one this year. I want to take
can displace 50%.
I wanna-
What you can see here is that column that's 100 nanomolar, one that's in the middle of this.
I've never smoked. I don't do, I'm like, I don't
So,
understand. I'm like, I'm
Some
telling
of
you right
these
now. I don't
compounds,
I
but
don't
not this compound in particular, Mitragyna, has high affinity for any single drug target that we're using.
have to do it. I'm
So
like,
even though it's claimed to be an opioid,
It says on my thing that
it does
examine my
interact with new opioid receptors and have opioid receptors, but
damn
also
it, no, they
interacts with
weren't.
serotonin,
They don't have to. There's
alpha
no.
one and alpha two adrenergic receptors,
I
dopamine
will do it
receptors,
because I get a lot
amongst a few others. So
told by
the
the by my
pharmacology is definitely complex.
when I shrivel up. I'm always paying attention.
The alpha adrenergic and
I hope
opioid
the microphone on this Zoom meeting isn't on, or else we're
Because this is an
talking about all
interesting
that.
therapeutic aspect where most opioid detox is treated with a partial opioid agonist like buprenorphine and also alpha 2 adrenergic agonists like lofexidine or clonidine, which were lofexidine was approved recently for opioid withdrawal treatment. And so we really were interested in, here's a molecule that's a partial opioid agonist like buprenorphine, but it also has this alpha adrenergic agonist activity as well, like lofexidine or clonidine that are used clinically as two different drugs, but now we got it all in one. So we're really excited about trying to move that forward. We also know that poison control center data that's been published from people that have called in and over-ingested material more represents stimulant overdose than it does opioid overdose. And so this is really interesting. These people are agitated, irritable, have seizures, tremors, cardiovascular complications. Very few of them have respiratory depression issues. So very much more like stimulant, cocaine, methamphetamine than it is opioid-like. So we wanted to look into that in detail. And just looking at analgesic measures in mice, we did a hot plate assessment with Mitragyna and a wild type mouse, and it gives full analgesic efficacy even as a partial agonist if you get a high enough dose. You can get 100% anti-nociception that lasts for quite a while, and eventually dissipates and goes away. We took knockout mice, mu opioid receptor knockout mice, gave them the same dose, absolutely no anti-nociception. Normal pharmacology would say, smoking gun, we got it. This is opioid, we're done. We pushed the dose higher. We pushed the dose higher in the knockout mice up to 200 milligrams per kilogram, and we started seeing anti-nociception return. And then we tried to reverse that with any opioid antagonist because they don't have mu, but they had kappa and delta. We didn't see any reversal with those. But what we did was we pre-treated those animals with yohimbine, which is alpha adrenergic antagonist, and we were able to wipe out all that anti-nociception that we saw in those knockouts. So now we believe strongly in this sort of dual acting theory of both mu and alpha adrenergic work. If you're interested, I'm going to just gloss over this slide real quickly. There was a study done by Smith, Klein, and French, published in 1972. The most complete work on Mitragyna that's ever been in the literature, as far as I'm concerned. This is a great paper for historical reference. And they were in full development of this as a new analgesic into the marketplace. They dropped it because it was similar to codeine. They knew it was reversible by opioid antagonists. And they didn't think there was a market to go up against codeine for anything else, particularly since non-steroidal anti-inflammatory class of drugs was starting to emerge. And definitely wasn't opioid, it was much safer. And so they abandoned it, and lucky for us, yes. There is a metabolite that is generated both intestinally and in the liver, and this is also generated in the plant material. We've never found it in fresh plant material. So when we've harvested leaves freshly off any trees, even if they're producing high amounts of Mitragyna, we've never found this compound in those trees, but we found it in many, many other marketed products, particularly in the older days. So we think it's a post-harvest artifact. It's an oxidative product that is produced by leaf drying and storage and whatnot that happens over time. The problem is, if you look at this table, When we put it through that 82 drug screen, it only hit mu delta and kappa opioid at 10 micromolar, most selective opiate that I've ever seen. And so we interrogated this guy, and of course, it's a high affinity opioid agonist. Very interesting, has a high abuse potential. This is work that we did with Scott Hemby to look at substitution for mitragynine in morphine conditioned rats that were trained to self-administer morphine. And so we basically took the morphine away, put mitragynine in there. They don't administer mitragynine, but they definitely administer 7-hydroxymitragynine at a much more lower dose. So this is just an equal substitute out for morphine. And this is a problem within the plant itself. On the good side, Jonathan Katz at the National Institute of Drug Abuse, one of the last projects he did before he retired was demonstrate that heroin intake in rats that were trained to ingest heroin could be reduced or completely blocked by pre-administration of mitragynine. And so this is interesting. This is an insurmountable blockade, not an antagonism. Because if it was an antagonism, and I don't have time to go into all these curves, but basically the inverted U-shaped curves you would see just shift to the right if it was an antagonist. Or if it was potentiation, it would shift to the left. But what's happening here is that curve is flattening all the way down, meaning those animals aren't injecting any material. So I'm going to skip this, but I'm going to show you that this metabolism does take place in the liver and in the intestine. And what we were able to do was dose intestinal microsomes and liver microsomes with mitragynine and quantify both mitragynine content that's left over time, as well as the appearance of the 7-hydroxymitragynine. And in both cases, we saw large increases in 7-hydroxymitragynine, which then we wanted to figure out what was going on. So we wanted to look at 7-hydroxymitragynine stability in human plasma and found that it's totally unstable in human plasma. And then we said, uh-oh, we can't do any work on this if it's not stable in plasma. And my postdoc kept telling me there's something else that's coming off the column at the exact same mass. And I said, well, what is it? He said, I don't know. We have to figure it out. So he hated me for this, but then he loved me when he got the cover of the Journal of ACS Pharmacology and Translational Sciences. But I made him scale up the plasma stability study and isolate out the unknown that was there. And what we found was a unique overproduced human metabolite that is produced in dogs, rats, monkeys, and mice, which is the smaller insert in all these curves at the very bottom of the screen here. Those are all the other species. The human is the only one that produces this at high levels. And so I made them figure out what this compound was. And it turns out there's one called mitragynine pseudoendoxyl. It's known in the literature. There was actually a fungal metabolite that was isolated when they gave mitragynine to a fungus, and this came out. Now, problem with this is it's even more potent than the 7-hydroxymitragynine. It is just as selective as a neuropeptide. The good side of that is it doesn't cross the blood-brain barrier very well at all. So it doesn't appear to have any kind of consequences in terms of when it's dosed. We've now looked at the generation of this in human plasma samples from actual human clinical trials, and we don't see levels that would do anything efficaciously at all. This was a study that we wanted to look at the consequence of the conversion of mitragynine to 7-hydroxymitragynine in vivo, and if that 7-hydroxymitragynine had any play in the activity. And I'll just make the long story short. The concentrations that get into the brain were never high enough to give analgesic efficacy when we did analgesic efficacy of 7-hydroxy by itself. So it seems to be non-consequential. We wanted to interrogate also what's metabolic pathways were involved. And so we did this by CYP profiling assays. We know that we've produced, I have it on here. Yeah, so 12 metabolites were identified just from mitragynine. in microsomes, and we identified 31 in hepatocytes. So the difference between microsomes and hepatocytes is you only have phase 1 enzymes in microsomes, you have phase 1 and phase 2 enzymes in hepatocytes, so it gives you more of a picture of what metabolites would be. So Mitragyna in itself, 31 metabolites. I told you there's 4 isomers of that compound in total. So now you're looking at 124 unique compounds that we have to detect and look for in the human trials. So I will also tell you that antinociception with the full product is reaches the ceiling. So you can never really get over this ceiling of maximal analgesic efficacy with Kratom Tea. So we make astronaut tea, I call it. We freeze dry it and then reconstitute it and deliver it to the animals. But it is having analgesic efficacy. We know that analgesia is mediated primarily through opioid receptors. So it comes back to that whole story about opioids again. And then just real quickly, I'll finish with showing you that it lacks a conditioned place preference. So we can train animals to be in an area where they prefer to get an injection of, where they got trained in an injection of morphine, so they prefer to hang out in that area when we put them back in that apparatus. Or if they're aversive to that, they'll hang out in the other side of that apparatus. And what we see here with lyophilized Kratom Tea at various doses is there's no preference. They don't care which side they go to. So there's no drug-seeking potential or aversive potential. We also saw very little sedative properties and very little respiratory depression. And again, I won't go into very much detail on those. We also know that it was very good at blocking naloxone precipitated withdrawal signs. So in animals that we habituated to morphine, then we precipitate withdrawal with naloxone. If we instead try to precipitate withdrawal with kratom, then we didn't see as severe withdrawal signs. Morphine's the one in red, various paradigms of Kratom Tea, and Kratom Tea all by itself, the last two. And you can see just a couple measures. One is teeth chattering, the other is jumping frequency. And then last but not least, what's the most important thing to take away from this? I mentioned there's 40 different alkaloids in the plant. When we do delivery of this plant material to the animals, we're actually seeing only 6 molecules ending up in the blood supply. You see 4 here on this slide. One of those is 7-hydroxymetragynine, which in our formulation was actually below the limits of quantification. So we know it wasn't present in the material we dosed them. So everything we see there is from metabolism. And then you can see mitragynin being the highest content there, which was the highest content in the plant material. Speciosialatin and corinanthidine. There was 2 other compounds, painanthine and speciogynin, that were there. So speciosialatin, mitragynin, and speciogynin are all 3 diastereomers of each other. So those are the 3 major compounds that we see. Speciosialatin is another opioid selective compound. It is the second most abundant in the profiles that we are looking at. So this is another compound that we need to be cognizant of and aware of, and maybe contributing to a lot of that pharmacology. And then the other 2 compounds, painanthine and speciogynin, their screens are almost identical. And then again, that's the one that just has that extra cyclic double bond that's reduced. These are actually high affinity 5-HT1A and 5-HT2B ligands, and also have opioid activity as well. And we did a quick study on these analgesic as well. These are reversed. They can produce 100% analgesia. These compounds that are primarily acting through serotonin receptors, if we use a selective serotonin antagonist, we can block or shift that analgesic efficacy to the right, showing that analgesia is primarily mediated through serotonin receptors. I mentioned a human pharmacokinetics study. The first one was published in March of this year. It was only 6 patients that completed it. They gave them a 2-gram dose. One of the patients that was excluded actually vomited right after their 2-gram dose. And so they couldn't be included in the pharmacokinetics portion because they don't have it in them anymore. So this was the first trial that got published. There's 3 trials that were just completed in Canada by Nutrisource. This is, you can Google it and see Nutrisource Kratom Plant Study Canada. They did a full plant material, they did an extract, and they did an isolate. And then finally, the FDA awarded a trial to Alta Sciences, which is planning is underway, and they're going to be looking at safety and tolerability study. And then after they show that it's safe, which is kind of a funny story, that FDA is actually wanting to show that it's safe so that they can then do a human abuse potential trial. It's strange to me, sorry. So the take home for all of this is there's this long history of safe use in the traditional manner in Southeast Asia with no deaths reported there for centuries, but it's been associated with a wide range of adverse events here in the West. The alkaloids are structurally very different from opioids and have very different pharmacologies and pharmacodynamic properties, pharmacokinetic properties. We know that products in the USA are not the same as what we see traditional products to be, and there's still lots of gaps around the science, but we still feel that it's a possible solution or part of the solution to help aid in ending the opioid crisis. So thank you for your attention. Sorry for our late start, which drove me late as well. So I'd be happy to answer any questions.
So if anybody in the chat has any questions, anybody watching on Zoom, you can drop a question in chat.
And if you have to go, I totally understand. Yeah, so the question is, do a lot of these other alkaloids play into the effect of the full product? Or is it just that one alkaloid? Is that what you're asking?
Well, that specific alkaloid is thought to be the primary like therapeutic constituent, so I'm wondering if any of the additional alkaloids that are in smaller quantities, like relative concentrations, affect the activity.
Yeah, so we have not really played around with the concentration yet. But what we have done is looked at each individual compound alone, and they definitely have different pharmacologies that I would say are probably making an overall contribution. But I want to also point out that I look at this thing as a symphony orchestra, and essentially what we're doing by looking at each individual alkaloid alone is plucking an instrument out of that orchestra and listening to it at full blast. And so we don't know what the actual contribution is when it's in the mixture with the full product. But that's part of one of the grants that we have, is actually to start taking out now that we know what's exposed to the animal when they consume the whole plant, now we can start taking out and making cocktails of these alkaloids and starting to look at which ones are creating which effects, which combinations and which ratios, all the fun things that now that we have the tools, we can start.
Got a question from the chat from Hamilton Podcast. Could you talk more about what we know regarding how 7-hydroxy and M-pseudo cross or don't cross the blood-brain barrier?
Yeah, so it's very interesting because we don't know a lot. We do know that 7-hydroxy is poor in crossing the blood-brain barrier when it's given alone. It crosses the blood-brain barrier more efficiently when it's given as Mitragyna. So the metabolite that's produced from Mitragyna, the 7-hydroxy metabolite, is getting into the brain at higher brain to plasma ratios than if you give 7-hydroxy alone. Now, what does that indicate? It indicates a few possible things that could be going on. One is there could be local metabolism in the brain of the compound. there could be some sort of transport mechanism that's being engaged, that's blocking or causing 7-hydroxy to accumulate more efficiently in the brain. We're not sure what those answers are. And what we've seen from dosing mitragynine, we can never detect pseudoindoxil, mitragynine pseudoindoxil, that even more potent compound, mainly because we're looking at mice and rats, and as you saw in that graph, they don't hardly produce. So we'll have to get that answer from some of the human pharmacokinetic data that's available to us. But if we dose 7-hydroxymitragynine alone, we can quantify in the rat and in the mouse mitragynine pseudoindoxil that's produced, and less than I believe it's 0.3% of that is actually getting into the brain. So it's a very minor amount of pseudoindoxil that's getting into the brain. And we have not done any studies where we've just looked at pure pharmacokinetics of pseudoindoxil to see if we deliver that as a single agent, how much of that is getting into the brain or not. So we haven't done that.
Two questions. One that kind of based on what you're just talking about. So pseudoindoxil 7-acetate, similar to the parallel between morphine and diacetylmorphine, as increasing permeability of the blood-brain barrier. Is that something, or even is that something along the lines of what you're playing with at all?
I'll just say from the get-go, we are not interested in developing an opioid for therapeutic purposes of pain. So if it's a partial opioid with different pharmacology and it works against pain, great. But I don't have an interest in trying to make these compounds that are very potent, more lipophilic and more brain accessible, because even if they do have better respiratory depression profile, they're still going to have high abuse potential. You're not going to be able to ever separate those two pieces out, in my opinion. However, there's companies that have been formed that are doing some of the things along the lines of what you're saying. I don't know. We'll just have to wait and see how the data comes out.
So a question from Brian Gallagher in the chat. Are most extract products you're seeing full spectrum or mitragynine only, or only a few alkaloids extracted?
Most of the extract products are just concentrated alkaloidal fractions. So just to give you a quick rundown on, if we get plant material, we have to extract and get the alkaloids out of that plant material. So there's a whole couple of weeks of process that goes through just to get rid of all the other plant material like chlorophyll, tannins, everything else. So we can get down to that alkaloid fraction. So there are products in the marketplace that have taken that piece out of the process. We actually use a lot of those extracts for isolating the compounds because it's more efficient and saves us 2 weeks worth of work. But those are what we see in those extract products. They're much more concentrated, or they've done heavy organic extraction processes to get those concentrations up. There are mitragynine isolate products that are out there. They're incredibly expensive from what I understand. I have not seen one in the marketplace specifically. Most of the ones that we see are 45% mitragynine enriched extracts. So. Hi, Brian, and hi, Hamilton.
Any other questions?
You said that alkaloid content, mitragynine content changes in the trees. Does total alkaloid content change? So is it just producing lower levels of alkaloid overall, or is it just the ratios that change? And then in line with that, can you trigger or induce, you were suggesting this sort of fungal purpose. Can you induce, by stressing the trees, alkaloid production in the trees that you have in the facility?
Short answer to all of that is yes, but we have not seen significant environmental triggers to really influence like greatly one way or the other in terms of alkaloid production. We know how to produce larger sized leaves, which then in turn gives us total larger biomass to collect, and then total alkaloid content goes up. And total alkaloid content is another major sticking point with me in the marketplace, because a lot of vendors will say, we have a total alkaloid content of X, but they're only measuring 3 or 4 alkaloids, and they just add those together and say that's their total alkaloid content. So it's really kind of a confusing piece of data that's out there right now. In the trees that produce lower amounts of mitragyna, we don't, I would say on a total weight percentage, we don't see a difference, but what in total content, but what we see is a difference in the expression of which compounds are there. So if it's low mitragyna, we see much higher production of other alkaloids. And we've been working with Sarah O'Connor, not from the Terminator, but from the Max Planck Institute in Germany. She's a biosynthetic plant person. They've been pulling out all the biosynthetic enzymes to really understand how the biosynthesis takes place. And what we're finding is there are some enzymes that if they are in low amounts in one chemotype, it doesn't produce mitragyna, and it produces another one of the diastereomers in much higher concentration. And so it may end up being that. And just to underscore what I said about not finding 7-hydroxy in live plant material, fresh plant material, they have not been able to find any oxidase that will convert mitragyna to 7-hydroxy in the plant genetic library that they've isolated.
Okay, since we have a class that should be coming in, we can leave any last questions for the 5 minutes as we all file out, and let's all thank our speaker one more time.